nanoHUB-U, online instruction.
Welcome back. We will continue in this lecture, Design Principles of Organelle Size, discussing what principles and design parameters determine the size of cellular organelles. So in this lecture, we'll talk about the second principle, Quantal synthesis, and we'll talk about the third principle, dynamic assembly, and go into a little bit more depth in a particular example that exemplifies this design principle and look at flagellar growth in algae. So in quantal synthesis, in this principle, the size of an organelle and that structure is determined by precisely controlling the amount of precursor material there is to start with. And so then the self-assembly dictates the size and number of organelles that are formed. So given some characteristic amount of material or number of monomer per organelle, those monomer pre-materials will self-assemble, determining the size and shape and number of the organelles. Now next, looking at dynamic assembly. In this principle, there's an equilibrium size of a structure that occurs when the rate of assembly and disassembly are in equilibrium. And often one of those two rates, either assembly of the organelle or disassembly of the organelle, depends on size, thus providing a feedback loop that determines that characteristic size of that organelle. And this is the principle that we're going to go into a little bit more depth in with an example. So flagella have served as a model system for dynamic assembly, dynamic balance control of organelle size for some time. So here we see some images of a biflagellate algae cell. You can see by biflagellate, I mean it has two flagella typically growing out. And here they've just been fluorescently labeled so that you can easily see the cell body and the two flagella sticking out from the cell. So the way these cells assemble and disassemble, forming their flagella, they undergo something called Intraflagellar Transport, or IFT. And that is the raw materials from the cell body, from the proximal end of the cell body, are transported down, and growth occurs at the distal end of the flagella away from the cell body. And so at that tip, there's assembly of material. And then for retraction of the flagella, which often occurs in many cell types for cell division and under different conditions, disassembly occurs at that tip as well. And again, the material can be transported back towards the cell body. So we have material going down to the tip, assembly down at the tip, and material coming back. And it's a balance of this assembly and disassembly that gives us our characteristic length. So in these cells, one of the things that you can do is to label these packets of material traveling down the flagella, these IFT particles during growth. And we can fluorescently label them. So you can see in this image now, you can see the fluorescence in these flagella are from labeling of these IFT particles. And people then can monitor the number and amount of IFT particle content in the flagella under different conditions and in different mutants in order to perturb and study this flagellar transport, assembly, and disassembly process. And one of the things that was found that if you look at flagella of different length as they're growing or as they're retracting, that the density of the IFT particles decreases as the flagella gets longer. And so if you look at this as the total number of IFT particles on a flagella, you'll see that it's constant. It's independent of length. So here in this figure C, we have the total integrated intensity. So this is looking at the fluorescence, which will be proportional to the amount of content, IFT content that's there, as a function of the length of the flagella. And you can see that that value is constant. So there's a set number of particles on the flagella, no matter how long it gets. So if there then is a constant velocity of transport, and if assembly is a transport limited process, in other words, it can only go as fast as raw material is delivered, then we would predict that the assembly rate will decrease as the length gets longer. And this is in fact what happens. So then if we write a simple kinetic model of flagella growth and write a function of the length of the flagella at dl dt, how that length changes with time, will simply be a balance of the assembly rates and the disassembly rates. So based on those biochemical studies that were done in monitoring the kinetics of the flagella growth, then we can write a term for the assembly of the rate equation, where the rate of assembly is going to be dependent upon that number n that we saw measured in the last slide, the number of IFT particles per flagella, divided by 2, because half the IFT particles are going toward the tip, half of them are coming back, right? So those total number on any given flagella that we count, about half of them are going to be going towards the tip for assembly. Divided by the length of the flagella times the length added per IFT particle or unit. So in other words, when that unit arrives and gets assembled at the tip, how much length is added to the flagella at that point. Times the mean, if we look at the mean transport rate, both anterograde and retrograde, in other words, going away from the cell body and back toward the cell body, then times that term, then we have our full term and representing the rate of assembly growth. And so now, just looking at the steady state solution of our dl/dt, we can predict a steady state length, which is going to be equal times the rate of disassembly times alpha velocity, number of IFT particles, divided by 2d. So now we've talked about flagella, algae flagella, as a model system for studying flagella growth. Flagella are also very similar and related to human cilia in human cells. And so I wanted to make this connection that this fundamental model system actually has very broad applications in a wide range of biological applications. So there are a number of human cilia diseases called ciliopathies. One example is polycystic kidney disease, or PKD. And this disease involves a human protein that's actually shared in common with the green algae IFT proteins. And it's defective. There's defective cilia assembly in this disease that involves the defect in this protein. And so I think this really highlights the value of studying fundamental design principles that cross a really wide range of species, from algae, which have important applications in bioenergy and bioprocessing, as well as in human medicine and health. So looking at a few other examples in humans and other mammals. So in our renal cells in the kidneys, so these renal cells also have a primary cilia that you can see indicated in this arrow right here. You can see the cell body back here in this electron micrograph, and you can see this primary cilia sticking out here. So these serve as mechanosensors that measure the flow of urine in the extracellular space past the kidney cells. Another important example, also where cilia act as mechanosensors in the human body, are in the auditory hair cells. And here, so these cells have little cilia that stick up out of the cell and connect with the tectoral membrane. And these cilia measure as sound waves come into the ear. You get deflection and bending of these cilia that transport signals into the cells of the outer hair cells. And this is a primary mechanism of auditory sensation. So next, coming up, We're going to go into organelle number. Again, we'll talk about dynamic balance plays an important principle for controlling organelle number as well as size. We'll talk about these design principles to control number, and we'll look at some specific examples in mitochondria and in chloroplasts.